Hey, Chuan, how
Hey,
are you?
Michael. Oh, you're still in the lab?
Yeah, I came back because it's either here or you hear my son screaming as he
Oh,
goes to bed.
I'm sorry. I'm sorry. If I know you have to go back to the lab, I'm probably gonna do it in the morning.
Oh, wait, there's options?
Yeah, there are options. I told you. You say in the evening's fine, so I think okay. No, thank you so much for doing this for us.
Yeah, no problem. Thanks.
Yeah, because um, I think the reason is that this platform is a lot of trainees from uh from Asia and
Hmm.
also from West Coast. So so I have this 12 hours difference. So it depends on the time zone. So if we invite speaker from West Coast, then normally we do in the evening because that's the only time they can do it. Probably
Oh,
like
okay.
6 p.m. for them. So for people in Japan or China, then it will be 9 o'clock in the morning for them.
Right.
So yeah, but anyhow, but thank you so much.
Yeah, no problem.
Yeah. So how's everything going in the lab?
Oh, it's going good. It's going good. We're just a new lab, so we're still well, I guess at this point, we're like established, so things are going pretty well. But
So
the
you
first few
got all the
months are
people you need?
what's that?
You got all the people you need?
Well, there's enough of us now, you know what I mean?
Okay.
Can
How
always
many?
use more. In fact, I'm advertising shamelessly in the talk.
Yeah, of course. Go ahead. That's the that's the point. I think particular for the juniors group, I think there are a lot of trainees looking for positions. So how many you have now in the lab?
Um, there's maybe like five, six of us, something like that.
Okay. Actually, I I was hosting a friend I think in this January he came to NIH give a neuroscience talk in NINDS. Yeah, NINDS. So yeah, he's really glad that your lab going pretty well. So
Yeah, yeah, no, it's good. Yeah, and things are moving, and
and the people
it's all
in the
very
lab now
exciting.
are PhD or postdoc.
There's some postdocs, a scientist, and some technicians.
What is exactly scientist?
Oh, so this is someone that's like a PhD level scientist, but not doing like an academic postdoc. So sort of like
Contractor?
a staff scientist. Hmm?
Contractor?
No, it's more like someone that doesn't like is not necessarily trying to get an academic job, but is like doing, like there's certain people with like certain skill sets that'll like just be in the lab and help with certain things.
But then, I know when I was in Brigham, there's no such a position. Everyone is like, except the technician, everyone is like on the contract on yearly basis, like instructor, postdoc.
Oh
For
yeah, no, it's a yearly basis, but it's essentially like someone with postdoctoral experience that's not like a postdoctoral trainee.
I see.
Like you know what I mean? There's people with
Yeah,
PhD.
I know. But
Yeah.
as I said, this sounds like this position used not existed in Brigham before. So when I was in Brigham,
Uh,
there's no such position.
I
So
think
either you're a postdoc or your instructor or your
well, you
technician.
must have known like Ed, right? I mean, that's basically Ed's job.
Ah, yeah,
Like
I
like
think
that
back
type
then,
of job.
back then there's very few positions like that. I think people are have issues like they trying to stay longer.
Hmm.
Like you said, they're not doing academia career,
Yeah.
just want to have a position,
Yeah.
like a contractor. But I think back then there's very few such options from I think hospital probably there's still possibility. From the medical school, it's almost impossible. I asked Isaac, he never has such some position like that
Well,
in
this one's a little different. This person's like running a core facility for us
I see.
that I basically run.
I
Um,
see.
so that's kind of like where the expertise is. But I mean, they're basically embedded in my lab to do
I see.
to
I
do
see.
that.
Okay.
Um,
Yeah.
yeah,
I think yeah, students could be a challenge at the moment.
yeah,
Yeah.
yeah.
There are
Student,
too
that's
few
it's
of them.
okay.
That's just the too few of them.
Yeah, yeah, of course, yeah, yeah. So I'm not necessarily.
You guys get any new faculty this year?
I'm trying to think. I am blanking if we
Well,
did.
VJ
I
got
feel like
this
there may have been a
new
couple.
center
Hmm?
ongoing. VJ has this new center ongoing. So I
Exactly.
figure there's money there.
Yeah, exactly. So I'm the first person as part of that.
Oh,
Um,
you are?
and yeah, yeah. And then
Then you probably get some
and
good spot, good meat from the
yeah, no,
first
yeah,
person.
it's super exciting. That's part of what this like core facility is like, it'll be kind of like application, like genomic applications for the institute.
But for this money, is there any new space or just everything in the Fenway building?
Uh, it's for now it's the same building, but there's new space.
New
There's like more space.
Okay, so I mean, but this is gonna be a center. I thought, are they gonna be like building some new place or as an
Oh, I
independent
don't know
place?
about that.
Okay. Well, yeah, I guess, yeah, Boston probably expensive. I
Yeah,
mean, 100 million, it sounds a lot, but if you want to build new things, then it's probably go pretty fast.
yeah, yeah, absolutely.
Yeah, but I mean, I think it's at least it's good for to support the new members in the. It's part of this the CND, is it? Or it's
No,
different?
it's independent.
I
But
see.
inter kind of like intertwined. A lot of the people have, multiple affiliations.
And then these things, is there any plan for the long run? Like how many faculties in total for this money to support?
I have no idea actually what the long-term plan is.
But the money is there, right?
Yeah, I mean, as far as I know.
Okay, well, okay, we'll see. I mean, it's been since it's announced, and now I think it's almost a year
Since
from
what?
the the BioLegend money.
Oh
It's
yeah, yeah. I think it was announced in last summer
yeah,
or
it's
something like
almost a
that.
year.
Yeah, yeah. Um, and yeah, no, so it's going. I think everything's are in like the early phases. We just had like a seminar series for one of the
I see.
programs. So there's like basically three major
Yeah,
programs. So we just had the first one. I think the second one is coming up next month. Um,
I see.
yeah,
Okay, cool.
so that'll be, so that it's sort of everything is like kicking off.
Yeah.
Yeah.
You want to give a try of your slides? We're gonna take a
Oh
look.
yeah, sure.
So it's okay to record, right? I just want to double check.
Yeah, that should be fine.
Okay, okay.
Let me see. So this isn't like a webinar, right? So this is like old-fashioned Zoom.
Yeah, old-fashioned Zoom. I mean, usually it's a public platform and it's open to there's no password so everyone can join. So usually I think it's depends on the topic and also I think most of a lot of I think half 50% of the speakers are senior speaker, 50 junior. We try to actually balance it. And then particularly we want to promote young labs and then try to see if there's any students which actually can be attracted to whatever subject that the speaker interested. And then, it's easy to communicate. And then, particular, I think although now people start to travel, but for the trainees, a particular trainees back in Asia, like in Japan or in China or in Korea, it's been still a challenge. So we are trying to actually organize a Cold Spring Harbor
Mm.
meeting in Asia. But even that, it's we have some challenge in terms of getting people to attend because it's a long distance, and then so but yeah, this platform will it's a lot convenient for the trainees to get know what your lab is doing, and then if any possible if there's a chance to join the lab or things like that. Yeah.
Cool. Yeah,
Um,
that's great.
so it was organized by a bunch of people, and then usually there's a co-organizer, Shale from Yale, together with me, but he's traveling today, so he wants to say sorry, and then but he's gonna get watch your playback on the web. So.
Oh yeah, no problem. So there's two of you guys that do this.
No, I think a bunch of us. I mean, sometimes
Oh,
if us are traveling, then there's third people. So there's a bunch of people from West Coast, also from East Coast. So we take turns. But most of the time is me. But if I'm traveling, then they will take over. So we actually have run this for over now close to 4 years, no missing one time. So no missing one time. So I think I'm pretty proud of that.
wow, that's
Yeah,
amazing.
so yeah, but that's teamwork. So it's as I said, it's usually take turns. If not me, then other people will join, and then there are always people will introduce or host the speakers, and then in the end, we usually have a quite casual discussion. So sometimes I just give you a warning, heads up. Sometimes the discussion could be longer than your talk. So I will try to not keep you in the lab too late today, I promise.
Yeah, yeah, you should have given me the 9 a.m. option. I didn't know it was an
I'm
option.
sorry, next time.
It'd be it'd be nice to.
We invite people back next time. Sorry, I should give you such options. Sorry.
I'd love to get home before midnight.
No, 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 no. It won't be that late. No, I promise. No, I won't.
Harvard,
I definitely will keep his things in time.
Harvard.
Promise. Your wife won't come out to kill you or anything like that.
Well, she'll be
Uh,
asleep. Don't worry.
yeah, yeah. Sorry about that. But I think it's 9 o'clock. Let's get started, shall we?
Cool. Yeah.
Okay, cool. Okay, I'm gonna just go ahead to record this.
Recording in
All right,
progress.
so it's 9 o'clock. Hello everyone. Welcome to our today's AMU Zoom seminar. So it's my absolute pleasure to welcome our today's speaker, Dr. Michael Wheeler. So Michael is really we don't have really too much overlap, but he's somewhere. I did my postdoc in Brigham Women's Hospital, currently is assistant professor in neurology. But there's a lot of fond memory back to Brigham. But yeah, we'll sure we're gonna catch up someday. So very, very brief introduction about Michael. So Michael did his undergraduate study in neuroscience in Johns Hopkins, and then continue with his PhD training also in neuroscience in University of Virginia. So after he completed a very successful PhD training, he actually moved to Boston and continue his postdoc training in neurology in Brigham Women's Hospital with Francisco Quintana, who also gave us a very nice talk last year. So from 2020, so Michael, because his really outstanding work, he was promoted as an instructor, and then following 2022, he was promoted as assistant professor, also in neurology in Harvard Medical School in Brigham and Women's Hospital. So Michael is really doing some very innovative and very exciting work. The main focus of his research program is try to study the neuroimmune circuits that control behavior and the neuropsychiatric diseases. His study is mainly try to aim to identify new mechanisms, machineries, and then develop new tools to study the molecular mechanisms regulating crosstalk between astrocytes, neural circuits, and the immune system, which I believe he's going to talk during his talk today. And then there's some really mind-blowing technology he developed with his colleague. I think it's super exciting. So in order to address these questions in his lab, actually using different approaches, including mouse genetics, genomics, and then mouse behavior, acutator technology, and the human samples, and also the new single-cell tools, which he developed to study cell-cell interactions. Their research program organized in different themes, which including but not limited astrocytes, immune interaction, how they control the CNS inflammation, and also try to study the new tools in order to understand astrocytes, immune cells interactions. And last but not least, his lab also interested in studying neuroimmune crosstalk in regulating behavior. So as I said, he's really doing some super exciting work, which I'm very much looking forward to it. Thank you so much, Michael, for joining us on this platform, and we are looking forward to your talk.
Yeah, great. Thanks so much for the invitation and the opportunity to speak to you today, especially at 9 p.m. So
Sorry.
make sure we'll get the global audience. So no problem. Um, so yeah, so as Chuan said, basically today I'm going to take you through a little bit of our work that we recently published, developing new tools in order to study interactions between the central nervous system and the immune system. Um, and I liken this a little bit to kind of returning to some of these principles of systems immunology to first understand how the components of each of these two complicated systems, the brain and the immune system, function, and then how to put those components back together to kind of get to something akin to physiology and disease. And so the main thrust of my work is to understand how the immune system and the brain communicate in health and disease. And I like to think of this as the kind of modern day mind body problem, which is we have these two very elegant systems, one of which is highly dynamic and in many cases transient, which is the immune system. And then another, which is the central nervous system, which is somewhat static and extremely delicate to investigate. And so the question is, in order to determine how these two systems interact, how can we sort of study the pieces of each of these in isolation first, and then figure out the mechanisms that regulate their interfacing? And so one of the problems that we've decided to apply this question to is multiple sclerosis, which is the prototypical demyelinating neurodegenerative disease of the central nervous system. And just as a brief primer, MS initially presents as a series of subthreshold clinical symptoms that are only detectable essentially by MRI. And then gradually, neurologic disability gets worse. It relapses and remits. And we believe that this is due to activation of effector T cells by a self antigen, presumably related to myelin. But what happens in multiple sclerosis in particular, and why this is an important problem, is as a disease gets progressively worse, there are actually no therapeutic options available, and the disease no longer remits, meaning patients develop a severe case of the disease and never recover. And one of the hypotheses that's put forward is actually this phase of the disease is driven by incompletely understood interactions between the nervous system and the immune system. So the question is, what are the cell types involved? What are the mechanisms? And how can we therapeutically target them to arrest the disease? And so clues as to what might be going on were actually put forward by Charcot himself in the 19th century, who reported that a newly classified type of cell, which was called nerve glue, which we now call astrocytes, actually displays a completely distinct morphology in patients with multiple sclerosis. And looking at a more modern day histological sample, you can see that these astrocytes extend foot processes directly to the vasculature, where they encounter immune cells that invade from the blood supply that extravasate into the central nervous system. And the important thing to remember about astrocytes is not only do they contact the vasculature, but they directly contact neuronal end feet and synapses. So in this way, astrocytes are very well positioned to sense immune-derived cues and relay this information within the central nervous system parenchyma. And so this is just a summary of how complicated the interactions between astrocytes and the immune system are. And so these are a number of different molecules and cell types that interact with astrocytes that we and others have recently defined. And so when we put this all together, there's not only astrocytes within the CNS parenchyma, there's also different classes of brain resident immune cells, as well as CNS extrinsic immune cells, such as T cells that can invade the brain. And so the question becomes very complicated in terms of what are the most potent regulators of astrocyte function that we could potentially target to arrest disease. And then the question becomes, are these interactions even important if we focus on astrocytes? And so these are some of the questions that we sought to address. And as I mentioned in the beginning, the way we really thought would be a useful exercise to understand these concepts is to really divide the problem into sort of bite-sized pieces, which is if we can figure out basic mechanisms that regulate astrocyte physiology with immune cells, let's say, basic mechanisms, can we then investigate those within the disease CNS and see what their impact is on physiology? And so, to really address that question, I'll tell you today about two complementary approaches that we recently developed to study this question. And as Chuan mentioned, these are the types of approaches we're extending in my lab to study other features of neuroimmune communication, such as those regulating behavior and neuropsychiatric diseases. And so, first, I'll tell you about this platform we developed, which we called SpeakSeq, which is microfluidic based and uses a coupling of cell pairs within a picoliter droplet environment. And we can make many thousands, millions of these droplets with cell pairs in them, and then rapidly prototype their phenotypes within the droplet. And I'll also tell you about a platform we developed, which is also microfluidically based, called FindSeq, where we can then take cell types of interest, for example, those that we found as importantly regulated in SpeakSeq, and isolate them out based strictly on transcriptional markers. So, in other words, you find a specific molecular class of astrocytes, you can enrich for it and define how it's distinct from other astrocytes and also other classes of cells. And so, first, I'll dive into this portion of the talk on SpeakSeq. And so, the way this platform works, or the way we hypothesized it in the beginning that it might be useful, is if we were able to take two distinct cell classes, one of which was expressing a randomly perturbed gene, and the other class expressing a state-dependent fluorescent reporter. And if we were able to couple those two cells together in a droplet, forcing them to interact, we could, in effect, see how changes in gene expression in one cell affected a phenotype in the partner cell. And so the idea we had was, let's make many millions or thousands of these droplets, culture the cells within these droplets so they're living. And then we use a droplet-based microfluidic sorter to enrich for the cells with a phenotype. And then the objective is to compare the perturbations that drive the phenotype we observe against those that do not drive a phenotype. And this was the platform that we sought to develop. And so the first thing we tested was, is it actually feasible to screen large libraries of genetic perturbations within distinct cell classes? And so this is just demonstrating that we are able to get a fairly noise-free encapsulation of cell pairs and droplets, showing that basically most droplets are empty, but then many droplets contain only one of the cell classes, and then a small fraction actually contains a pair, and then very few droplets will actually contain more than a pair. And so we're able to then just microfluidically through cell concentration load these droplets with the requisite number of cell types to prevent false positives from multiple cells with perturbed genes affecting a specific phenotype that we would otherwise miss. And so we used a microfluidic device to encapsulate cells, which can basically take two distinct populations through these channels. And then as an output, you can see that we're able to put many of these cells in droplets as an output. Chuan, just wanted to check, is my internet breaking up?
No, it's fine.
Oh, okay, thanks.
Yeah.
And so what we were able to take advantage of is a droplet microfluidic sorter that we developed, which is capable of detecting spectrally distinct dyes. And so what we decided to do was we labeled each cell class with a unique dye. We labeled droplets themselves with a unique dye. And then we also were able to gate on a fluorescent signal that's emitted if a cell exhibits a certain phenotype. And so the idea was we can gate on only droplets containing pairs of cells that express a phenotype. And by isolating the guide RNAs that are expressed in the cell type of interest in those droplets, then we can figure out a class of genetic perturbations that affect a phenotype of interest to us. And so we performed a number of controls, making sure that when we actually cultured these cells in droplets, that each droplet was itself independent of another. And so what we first did was we optimized some of the media that we use to prevent merging of droplets into these kind of large blobs. And so by simply removing phenol red, we can make many of these droplets that are stable over time at physiologic temperature. And then we also performed a mixing experiment where we loaded two independent populations of droplets with distinct dyes. Then we cultured them together and we quantified how many of the droplets contain both dyes. And we see that only about 1 in 2,000 events, this droplet in the middle here, actually contain both dyes. So by and large, the droplets do not mix contents and they do not merge over time. Importantly, we validated that when cells are encapsulated in these droplets, that they are viable up to a certain point after a certain amount of time. So um, we quantified this over time, and we find that at around 1 day, we still have a reasonably high number of living cells, so greater than 80%. Um, and in related studies, we found that at 24 hours, cells are able to exhibit phenotypes associated with gene expression changes that turn on genetic reporters. And what's important actually about this experiment is because of the way that the droplet microfluidic sorter is constructed, we're able to gate out droplets with dead cells in them because the same dye signal is now diffused within the droplet instead of focused within living cells. And so we can simply gate out peaks that don't reach a certain amplitude within a droplet to essentially eliminate cells that are dead and have simply leaked their contents. And so finally, one of the last things we sought to validate is can we actually induce stimulus dependent activation of these cells? And so astrocytes are sensitive to the innate site against TNF alpha and IL-1 beta. And so we did a number of different controls showing that if we basically pump TNF and IL-1 into the droplets, we can dose dependently activate astrocytes that are expressing a reporter of NF-kappa B. And so I think the schematic's not shown, but basically in these mice, when NF-kappa B gets turned on, it gets recruited to responsive elements and it turns on EGFP. And so these cells from this transgenic line will start to become green. And testing this in droplets with or without companion cell types, we find that basically astrocytes will only activate this inflammatory pathway if they're co-cultured with a cell type such as microglia that are themselves activated. And so just going through each of these controls here, if we culture astrocytes that are stimulated at a sub-threshold level, meaning GFP is not turned on, which is just about here, we see that there's really no activation of the cells in droplets. Similarly, if we culture them with activated microglia that do not have randomly perturbed genes, so just control targeted microglia, there's similarly very little activation. However, when we treat microglia with a CRISPR targeting viral library, we can see that genetic perturbation of certain genes is sufficient to activate the reporter in an astrocyte that has been paired with microglia. And just to say, so the remainder of the experiments I'll show you using this speak-seq approach are using astrocytes that are stimulated at this sub-threshold level. And the reason we chose it is because our goal was to unmask negative regulators of astrocyte pro-inflammatory responses because these have been shown to be important in multiple sclerosis. And because we thought that this would be a great starting point to just tip the balanced just so that NF-kappa B would be turned on in these cells. Because actually, if you don't activate these cells at all, then there's few genes that would actually just drive the activation of NF-kappa B at baseline. And so this is the paradigm we basically decided to use. And so finally, I'm just showing you some live trace data from our sorting where we can dissect out events coming through our droplet microfluidic sorter in real time, where each of these low intensity purple peaks is a droplet, each of the red peaks is a partner cell type, a purple spike is a partner cell, and then the green spike is an activated cell. And so you can see we can screen many of droplets um, uh, across time. So we're able to really screen thousands of genes at any given moment. And we believe, this system can go genome wide if you have enough cells. And so this is just the visualization of sorting cells that have been encapsulated in droplets. And so we used a sorter that was designed by Ian Clark, who's the co-first author on these studies. And basically, he designed it such that whenever we give the sorter specific criteria, it can pull droplets in towards a specific gate. And then these gates go out into independent tubes, and we can read out the transcriptional changes in each population. And so to get to the actual experiment, what we decided to do was randomly transduce microglia with a genome-wide CRISPR-Cas9 library, such that only one gene would be perturbed in an individual microglial cell at any given time. And in parallel, we harvested astrocytes from a reporter line that I showed you that expresses EGFP under control of activated NF-kappa B. And our goal was to randomly pair these astrocytes with perturbed microglia with the goal of finding perturbations that in microglia that would drive NF-kappa B activation in astrocytes, in effect telling us microglial-derived negative regulators of astrocyte pro-inflammatory responses, which have been shown by many groups to be crucial in shaping neurodegeneration and inflammation. And so this is just an example droplet cytometry plot where first we actually gate on cells containing an astrocyte that has been activated. And then we also gate these cells on droplets that also contain microglia. And so this is essentially our positive fraction. And anything that doesn't meet all of these criteria gets sent to the negative gate. And so now we're able to really determine what are the molecules that, when expressed in microglia, limit these astrocyte inflammatory responses. And so we tried a number of different controls just to validate that when we're sorting these droplets, we're detecting physiologically relevant interactions. And so this is just quantifying the activation by droplet cytometry of different conditions. So if we transduce either microglia or macrophages with a non-targeting lentivirus that also delivers Cas9, there's really not much induction. However, when we target each of them with this Brie library, which delivers random perturbations to specific genes, we can see that there's a significant induction of astrocyte inflammatory responses. We see that we're isolating a number of droplets commensurate with a single perturbed cell per droplet, since there's a nice correlation between the number of guides that we detect and the number of positive droplets that we isolate. And then lastly, we believe these hits are physiologically relevant because within the positive fraction, there are comparatively few non-targeting guides from this genome-wide library expressed at high levels, whereas in the negative fraction, there are many. And so we believe this is a faithful representation of astrocyte microglia communication. And so using this platform and analyzing astrocyte microglia crosstalk, we analyzed about 1,500 genes that we believe to be hits from this screen. And we cross-validated this list using a bulk RNA-seq preparation of microglia that were treated in the exact same conditions. So essentially filtering out genes that were only lowly expressed by microglia, basically just enriching for genes that are well known to be expressed by these cells. And when we do this, we actually see expression of molecules that we know are important for anti-inflammatory function, including IL-10, the IL-33 receptor, and other molecules. And then finally, we wanted to validate that other groups had detected these hits previously. And so we cross-referenced our data with four other published studies analyzing microglia by bulk and single-cell RNA-seq. And of the greater than 1,000 hits that we detected, only one was unique to our speak-seq screen. Other than that single hit, other groups had detected all of these molecules, and almost all of them were detected by all of the groups. And so we believe that these were valid interactions. And so from there, we wanted to investigate the biology that we potentially uncovered. And so we first analyzed ligand receptor pairs that might be mediating crosstalk between astrocytes and microglia. And so we searched the genes uh, that, when perturbed in microglia, induced astrocyte responses that were filtered against all of these other data sets. And we looked for specifically secreted factors. And we settled on these four because we thought that not much had been shown about their crosstalk with astrocytes. And so each of these genes signals to a specific receptor, which is expressed by astrocytes. And we ultimately settled on this interaction between AREG and EGFR. So AREG stands for amphiregulin, which is actually a growth factor that binds EGFR, which is the epidermal growth factor receptor. These are well known to be anti-inflammatory in other contexts. And so we thought it was a potentially interesting mechanism because AREG is also state dependently induced in cells in the context of inflammatory responses. And so we just validated using in vivo preparations of astrocytes and microglia that these molecules are expressed by each of these cell types. And we see that AREG is expressed by microglia. It goes up in response to a preclinical model of MS called EAE. And EGFR is highly expressed by astrocytes. And I should say, we also performed a small in vivo screen by Perturb-seq to really narrow down this search to AREG. And so our next step was to validate whether or not AREG actually serves this an anti-inflammatory role in vivo. And so what we did is we transduced mice with a lentivirus, delivering a guide RNA against amphiregulin and also co-expressing Cas9 under a microglia-specific promoter. We performed an ICV injection, and we induced EAE in these mice, which is an active immunization against a myelin peptide that induces progressive ascending paralysis. And so basically, the higher the number on this y-axis, the worse the disease is, the more paralyzed the mice are, and the x-axis is time. And so what we see is that when we get rid of AREG, the mice do significantly worse than controls. When we analyze the astrocytes by RNA-seq, we see that astrocytes, when AREG is perturbed, upregulate a number of chemokines and cytokines that induce pro-inflammatory responses and can be linked to neurodegeneration in the CNS parenchyma. And they downregulate homeostatic markers that are protective. And just validating this on a pathway level, we see enhanced activity by NF-kappa B, which validates the original speak-seq paradigm, which was using NF-kappa B reporter cells. And so we knew that AREG was protecting the CNS from a hyperinflammatory response, but the next question is how and what's the role of the astrocyte? And so we know actually, so this is work from David Artis's lab. So we know in the gut that amphiregulin production in ILCs is controlled by the alarmin cytokine called IL-33. And so in the context of tissue damage, IL-33 is released from dying cells, and it can induce, in some cases, protective mechanisms that limit inflammatory damage. And this was an example in the context of gut inflammation. Anna Malofsky's lab, for example, showed that similar properties hold during development, which actually prevent behavioral deficits driven by IL-33. And so we thought that IL-33 might be a putative regulator expressed by astrocytes that limits these inflammatory changes by potentially controlling amphiregulin production in microglia. And so to test whether this might be the case, we actually reinvestigated a previous data set that we published of astrocyte microglia interactions in the context of autoimmunity. And not to get into the details too much here, but we basically investigated cellular networks consisting of astrocytes and microglia. And we asked, what are the potential regulators of EGFR positive astrocytes that communicate with AREG positive microglia compared to microglia that express another EGFR ligand called TGF alpha. And what we stumbled upon is one of the top predicted cytokines that controls these cells is IL-33, suggesting that IL-33 might be a key driving factor in establishing the anti-inflammatory properties of AREG in the context of autoimmunity in the CNS. And so we wanted to validate whether or not this was the case. And so we stained the naive or EAE spinal cord for IL-33 and GFAP. And we see that there's a significant induction of IL-33 expression in astrocytes. And we know that in the context of autoimmunity, astrocytes can apoptose and release their contents, suggesting that astrocyte-released IL-33 might be acting on IL-33 responsive microglia that then turn on AREG to drive this anti-inflammatory circuit in response to damage. And so to test whether this is the case, we actually bred, together with Vijay Kuchru's lab, astrocyte-specific IL-33 knockout mice, and we induced EAE in those as well. And we see that if we get rid of IL-33 in astrocytes, the mice do significantly worse. And then doing the converse experiment that I showed you before, so in other words, analyzing microglia from an astrocyte-specific knockout, we can see that there's actually a significantly diminished production of amphiregulin by microglia when IL-33 in astrocytes is deleted. And I should say, I don't think I have time to show it, but we also deleted the IL-33 receptor in microglia, which phenocopied this data, suggesting that this is the circuit mediating these effects. And we think it's always important to validate these findings from preclinical models in human samples. And so in collaboration with Alex Pratt and Jack Antel, we stained MS patient CNS tissue for microglial markers such as TMEM119, as well as AREG, and we see a significant elevation of AREG in MS lesions, which are the predominant sites of early damage in this disease. Indeed, we actually see an elevated IL-33 signature in microglia from a data set that we reanalyzed that was published by Marco Prince. So the question to us, of course, was if this program is elevated, then how might it be dysregulated in disease? And what's interesting is that it turns out that in MS patient astrocytes, EGFR is significantly downregulated, suggesting that although there might be increased AREG in response to this IL-33 dependent damage, the astrocytes are less sensitive to this program, which may contribute to disease. And so this is work that we're interested in following up on to see sort of the temporal dynamics of this system and really what other cell types might be involved, because neurons also produce IL-33, and of course they degenerate in multiple sclerosis. And so just to briefly summarize this part of the talk, so I showed you that we developed this platform, which we called SpeakSeq, which is a droplet microfluidic platform to screen mechanisms regulating cell-cell communication at high throughput. And using this approach, we uncovered that microglial-derived AREG limits inflammatory astrocyte responses that are controlled by NF-kappa B by activating EGFR. And this circuit is controlled by IL-33. And so now that we have, this platform to study individual populations of cells and the mechanisms that regulate them, the next question is, what if we really want to dig deep into one specific cell population that we've uncovered? So for example, AREG expressing microglia, or EGFR expressing astrocytes. I mean, it's challenging to do this with conventional technologies because you need a surface marker that works well, or you need a genetic targeting tool where you can access them. And these aren't always available. And so this is one of the central problems that motivated us to develop this tool called FindSeek. And so the motivation actually came from a previous study that we had, where there's a state-dependent activation of a transcription factor called XBP1, which upon its activation gets transcriptionally spliced into an active form, which we found that when it's elevated in astrocytes, it turns on a number of cytokines and inflammatory factors that can contribute to CNS inflammation. Um, Now, we knew that XBP1 in astrocytes is pathogenic because through both genetic perturbation using viruses and also transgenic animals, if we got rid of XBP1 in astrocytes, it's significantly beneficial in disease, and this is similar to what others have shown in macrophages and astrocytes in other contexts. And it also decreases the recruitment of inflammatory monocytes to the CNS during autoimmunity. Um, what's important is that we also detected this pathway is relevant in human samples. And so if you look in active lesions of MS patients, you can see elevated induction of spliced XBP1, which appears to be predominantly expressed by astrocytes in these lesions. And so we know it's physiologically relevant, and the immunostaining is shown below. But this specific problem we ran into is twofold. First of all, as I mentioned, XBP1 is transcriptionally spliced. And so this splicing event is difficult to detect by single cell RNA-seq, depending on where it is in the transcript. And so as a surrogate, we looked downstream of XBP1 activation for its target gene called EDEM1, which if you have a mouse that is a knockout for XBP1, you don't get any EDEM1. And so we thought that this might be a nice surrogate marker to show XBP1 activity, and it would be a logical choice because it's downstream, it would represent amplified detection of XBP1 activity. But again, we ran into a problem where when we analyzed astrocytes that we captured by single cell RNA-seq for this transcript, we only detected 14 cells out of about 1,200 that expressed it. And so we really encountered a problem, which is we know these cells are important, but we can't access them genetically. And so sort of how can we solve that? And we turned to microfluidics once again, where what we developed was a platform where a complex tissue can be encapsulated uh, within an agarose gel matrix that captures its RNA, but also its DNA. And each cell can be lysed in an individual droplet. The RNA can be reverse transcribed within the droplet. And then basically, you can pump in TACMAN reagents for a transcript of interest. So in general, we were multiplexing two transcripts. And so basically, you can take a cell type of interest and look for a molecule that is of particular importance to you. And then through saturation PCR, these droplets containing cells expressing a specific marker will fluoresce. And then using the same droplet cytometer setup that I showed you for SpeakSeq, we can enrich for these transcriptionally defined markers. And so what I think is important here is that now leveraging the power of genomics and single cell RNA-seq, we can enrich for populations that we discover using these tools without having to generate, cumbersome transgenic animals or developing antibodies or proteomic tools to identify molecules that they might express on the cell surface. And so using this approach, we developed it where it can be in sort of a pseudo bulk, which is about 100 drops or 100 cells, or we could sort individual drops into the wells of a 96 well plate and do single cell FindSeq. And this is just a representative schematic of the sorting device. And so you can see you kind of have this droplet that's a bit of a needle in a haystack. But after sorting, you can enrich for this very specific population of interest. And I should say, actually, this approach, um, developed by Ian Clark, also captures DNA within the agarose matrix. And so he used it in a parallel study that captured HIV provirus. And so he was able to enrich for latent uh, viral reservoirs in HIV. And so it's a versatile tool that I think has many applications. And so just to remind you of how this XBP1 processing works. So once the transcript is spliced and it's translated, it becomes a potent transcription factor that can activate a host of target genes, including this gene called EDAM1. And so we designed our FindSeq assay to uncover EDAM1 expressing astrocytes. And so we have an astrocyte marker, aquaporin 4, and this marker of XBP1 activation, EDAM1. And we can see, consistent with what we found before, spliced XBP1 goes up about tenfold in EAE. And when we enrich for these cells, performing FindSeq and then performing RNA-seq on these cells, we can see that we actually detect substantial number of transcripts comparable to other leading approaches, such as single-cell RNA-seq or SMART-seq. And so we basically have the sensitivity to detect the entire transcriptional regulation of a transcriptionally defined population of interest. And so we isolated out astrocytes expressing or not EDAM1 in the context of naive mice or EAE. And what we find is we can see even within these narrowly defined cell pop, we can cluster cells into distinct classes. And we found a population of aquaporin expressing, EDAM1 expressing astrocytes that we analyzed by transcriptional network analysis. And we saw that in the genes that are downregulated in these XBP1 expressing astrocytes, one of the top ones was encoded by a gene called NR3C2, which encodes a nuclear receptor transcription factor known as the mineralocorticoid receptor, well known to antagonize NF-kappa B activation and responsive to steroid hormones. So it's well known to be anti-inflammatory. And so it suggested to us that XBP1 and the mineralocorticoid receptor might mutually antagonize one another in the context of CNS inflammation. And so what we found is if we treated astrocytes with inflammatory cytokines in vitro and blocked the mineralocorticoid receptor, we can actually drive increased splicing of XBP1. So essentially, mineralocorticoid receptor loss of function boosts XBP1 activation. And we found a comparable phenotype when we inhibited the MR on cytokine and chemokine production. So essentially, it boosted the expression of all of these molecules. We did the opposite experiment by perturbing XBP1 and analyzing the activation of the mineralocorticoid receptor. And what we see is that if you knock down XBP1 and then perform a TAC-seq, you can see elevated chromatin accessibility of NR3C2, as well as its cofactor called NCOR2, which is actually a co-repressor that limits the expression of certain loci that NR3C2 is recruited to. And so using our single-cell FindSeq data, together with our bulk FindSeq data, we found that NCOR2 is actually one of the top cofactors that was downregulated when we perturbed NR3C2 in astrocytes using a comparable approach. And basically, it was predicted to negatively regulate the XBP1 driven signature to a comparable degree as the mineralocorticoid receptor, which suggests that they were working together. And so in vivo, we tested this idea. I should say that we knocked down the mineralocorticoid receptor and it exacerbated EAE, and also it also boosted XBP1 activity, but I just don't have time to show it. But we also knocked down NCOR2 to determine if it phenocopies the NR3C2 knockdown. And what we see is that if you ablate NCOR2 specifically in astrocytes using an astrocyte-specific promoter delivering Cas9 and then co-expressing a guide RNA against it, you can you actually downregulate a host of anti-inflammatory molecules and boost the expression of chemokines and other inflammatory factors. And also, when we look at XBP1 activity, we can see that loss of NCOR2 elevates XBP1 activation, similar to what we found using the other loss of function approaches. And so, just to summarize this portion, we found that using FindSeek, we uncovered that the regulation of XBP1 expressing astrocytes is controlled by the MR, and these cells are important for multiple sclerosis patients. So, in other words, they're elevated in MS patient lesions, and so this targeting the MR NCOR2 pathway as a potentially immunoregulatory pathway might have therapeutic potential, and really because there's not many tools available to study these molecules, we believe FindSeek is a unique approach to kind of uncover features like this, but also other molecular mechanisms. And I guess I probably should have said all this first, but the two papers I told you about today were done with my co-first authors, who Ian Clark, who I mentioned, who now has his own lab at Berkeley, and Hung Yoon Lee, who is still a postdoc in Fran's lab, but I should say he's on the job market, and he just had a great paper studying immune memory in non-hematopoietic cells, so astrocytes, in the context of neurologic diseases. And so, just to circle back to this theme of how we can sort of deconstruct cell circuits and the molecules that regulate them in the context of health and disease, my lab is very much interested in dissecting these problems at a circuit and systems level in the context of behavior, inflammatory diseases, and addiction. And so we're very interested in how the immune system might be facilitating some of the changes that we observe in these disorders, and how we can potentially target them using unique therapeutic approaches. Um, and so with that, I'm happy to conclude and thank you all for attending this talk. And thank you again, Chuan, for the invite. And I'm happy to take any questions. Thanks.
Recording
Hey,
stopped.
so thanks a lot, Michael. Well, it's never get old, you know, every time I hear this talk, all the technologies, I mean, it's always blow me away. But yeah, so we open the floor for the questions. If anyone has questions, please type in the chat box or just raise your hand. I think I'm gonna start to kick off. Actually, first is a technical question. So when you do the speak seek, in the end, when you actually sort for the sequencing, what's the number of the cells for the final sequence?
Yeah. So we were able to sort, I think, I forget the lowest number, but it could have been hundreds of droplets. Um, and so, yeah, I think it was hundreds of droplets on the low end. And so what we're isolating is DNA from these cells because the lentivirus stably incorporates, um, and that's what we amplify. So we basically used the existing protocols for isolating guides from stably transduced cells, and we're able to optimize it for low quantities of material.
So it's just it's you can obtain the information from like less than 1,000 cells.
Yeah. I mean, hundreds is what we're able to isolate in some cases.
Okay. Then it's actually going to this AGR ARAG story you talk about. So when you actually do the interaction between the ligand and the receptor, so my question is this something because when you actually do the cell-cell interaction, I assume that this is somehow based on the naive state instead of the inflammatory state, right?
In SpeakSeek.
In the speak-seq, because you just mix the astrocyte and the microglia together, but this is to mimic what happened under homeostatic stage in the brain, or you are thinking this is somehow like the EAE stage.
Yeah, I think I didn't explain that clearly enough. So what we did try to do is mimic an inflammatory state, and so in all cases, astrocytes were stimulated with a subthreshold dose of IL-1 beta and TNF alpha. Subthreshold meaning that it doesn't turn on the NF-kappa B driven reporter. So the
Hmm.
cells are sort of teetering at the brink of activation.
I
And
see.
then in parallel, the microglia are pre-treated with LPS,
I see.
and they're overnight, and then all this gets washed out before you encapsulate the cells into a droplet. And we know that if you culture the cells alone, there's not any activation. If you culture them without even both activated, but without a genetic perturbation, there's no activation. And so that's how we decided on this system.
But
So we do
then,
think it mimics EAE.
usually I noticed that you guys now actually using the virus approach to knock out the specific gene in the astrocytes or microglia. So I'm just wondering when you actually induce such perturbation in the CNS, then the brain stage is somehow mimic the EAE, or it's as far as you knock it out, does these mice have any phenotype even without induction of like a mob injection?
Oh, well, you mean in just like an inflammatory state?
Right. Yeah.
Well, yeah, I think it's a good point, but what we found previously is if you compare any inflammatory pathway of
Hmm.
a virally transduced mouse, one of which has EAE and one that doesn't, it's not
Hmm.
even a competition about the how dramatic the inflammatory change is in the EAE mouse. So we think that first of all, if there are inflammatory changes, the those are inferior to those induced by EAE itself. And
Hmm.
also, all the mice are transduced. So the control mice are transduced with a non-targeting guide. And
When
so
when you induce the virus, you do IV injection?
we do ICV, so directly into the CNS.
I see. Okay. And then the coverage of the infection was sufficient. I mean, I'm not into that much technical details because I'm always curious when you do the infection with the virus, how how many astrocytes or microglia can potentially be knocked out by your guiding RNA.
Yeah, exactly. So I think we routinely see around 50%-ish knockdown of protein.
Okay.
Um, and we do inject quite a lot of virus to try to cover as many cells as possible, and we do it into the ventricles to try to diffuse throughout the CNS.
And
And we can
then is there
detect it
any
in the spinal.
somehow preference of these viruses can go? Because I'm actually thinking whether when you inject, when you do some injection, then the virus tend to go specific area instead of somewhere is more, somewhere is less. Is something like that happen?
Yeah, I mean, we inject into the ventricles, which then kind of are able to uh, uh, propagate the virus to multiple different areas. I think the region that we target the least is the cortex, but that's okay with us because the pathology is predominantly not in the cortex in this model, and we
I
do
see.
see it in the spinal cord where there is pathology, but
I
also
see.
in uh, uh, structures, beneath the cortex.
I see. Then regarding to the biology, because when you actually identify this interaction between the AR, um, AREG, ARAG versus the EGFR, but when you do the ARAG knockout from the microglia, although you see the phenotype, but I mean to be kind of consistent with your sequencing data, I think it's hard to say that the reason the knockout of HR ARAG is because the astrocytes responding to this. Because in the CNS, there's so many other cells potentially can perceive the ARAG signal, not only astrocytes. How can you demonstrate that this knockout on the ARAG in the microglia is eventually cause the EAE difference is because the responding response from the astrocytes?
Yeah, it's a good question. What I can tell you is that when we knock down EGFR and astrocytes, it more or less phenocopies the AREG knockdown.
Hmm.
Um, and you didn't ask, but I can say that, in terms of whether or not microglial AREG is important enough, we did a bunch of controls to determine whether AREG produced by other classes of cells, like Tregs or I think we even did it. We did, yeah, we did a germline knockout. We did a T cell, a CD4 T cell knockout, and we also did a bone marrow chimera. And basically, anytime you can target microglia, that's when you recapitulate the phenotype. But if you target another cell, you don't. But in terms of its specific effects on astrocytes, it's true that of course it might act on other cell types that express the EGFR,
Mhm.
but we do believe it's a phenotypically important cell circuit because it changes EAE severity.
But regarding to this ligand receptor interaction, I remember, I mean, I don't exactly remember if you actually described in your talk that whether AREG can somehow, when it's actually stimulated the EGFR astrocytes, whether that can impact the EGFR expression level.
Oh, whether AREG drives EGFR itself?
Right, right.
I think it does. We have some preliminary data that suggests, like in other contexts, getting rid of microglial AREG influences EGFR expression in astrocytes. But yeah, I don't. We didn't directly measure that in this study.
I see. But eventually, when you actually say the IL-33 upstream of AREG,
Mm.
so again, I still have this question that whether IL-33, I mean, to target the microglia, is that something specific for AREG or something I figure it's probably do a lot more than the AREG, right?
Oh, you're asking if
Upstream.
IL-33,
Yeah.
yeah, yeah, yeah. So the one thing is microglia are the predominant expressors of ST2 in the CNS. Now, of course,
Ah,
there will be infiltrating immune cells in
mhm.
EAE,
Yeah,
which will express ST2.
right.
Um, but because microglia and astrocytes are in such close proximity in general, um, we think that at least a substantial portion of those effects might be on microglia themselves.
I see. So you think I see. So you think that but IL-33 could potentially um release from any cells.
Absolutely, yeah, absolutely.
Yeah, I see. So when you actually induce the CNS inflammation, that a different source of the cells which actually release of IL-33 and then mainly focus on the microglia, that's what you mean?
Uh, yes, yeah, exactly, exactly. I mean, we focus on microglia, but I mean, there's no reason why other cells that express it wouldn't respond. Um, but what I'm saying is that within the CNS, microglia are the predominant cells that express the receptor for IL-33.
But as far as I can recall, I think, like you said, during the EAE, when the T cell infiltrate, they also express ST2. And
Yeah,
then,
yeah.
but you don't think the T cell can contribute to the ARAG production?
Oh, that's well, I mean, we actually did do that experiment. So when we knocked out AREG in T cells and induced CAE, there was really no difference.
Oh, I see.
Yeah,
Cool.
yeah.
Okay. Well, I mean, I don't see any other questions in the chat box,
They're
so
giving
you
me
can
a
go
break
home
because it's
early.
late. I love it.
Uh, well, but anyway, so I think Michael actually presented a lot of cool technology today, and then I think probably take some time to digest. But if anyone in the audience has questions for Michael, I think he'd be happy to take questions by email. And also, I think Michael is actively recruiting. So like you see, he's doing really cool stuff in the lab. And anyone from the audience interested, I think just reach out. And then, I think, yeah, his lab is doing very well. So, but with that, Michael, thank you so much to present today with us and stay late. But as I said, I'm not going to keep you anymore. So thank you so much. Hopefully, see you soon in person. I mean, if you drop by an edge, let me know.
Yeah, yeah, yeah. Thank you so much. This was
No,
great.
yeah, sure. Thank you. So stay late for us. Go home now.
Yeah.
Thank you so
Take care.
much.
Thank you
Thank you.
again.
Take
Thanks,
care.
everyone.
Yeah. Bye bye, everyone. Bye, Mike.